Simple organisms. On the next 4 pages, you can find out more about looking at very simple living things, lower organisms. They do not have the same structure of cells as true plants and animals, higher organisms, and are normally thought of as being neither plants nor animals. Bacteria. Bacteria are very tiny organisms made up of just one unit, which is simpler than and very different to a plant or animal cell. They live and multiply anywhere they can find a source of energy to live on. Some live on or in other living things, either being harmful or causing diseases, or useful, for example, protecting against more harmful bacteria. Most bacteria, though, live on or in dead plants and animals, and their actions cause decay or rotting. This is very important. See Decomposing, page 18. We all suffer from diseases caused by bacteria, but we also have useful bacteria living in our bodies, like those which help us digest food. Spherical bacteria are called cocci, singular coccus. Rod-shaped bacteria are called bacilli, singular bacillus. Looking at bacteria. Groups or colonies of bacteria can be seen easily with low power. A single bacterium is too small. Bacteria form colonies very rapidly, multiplying by splitting in two, as long as there is enough food. You can grow colonies of harmless bacteria on jelly in shallow dishes with lids. Petri dishes, see page 46. For about 12 dishes of jelly, you will need a vegetable stock cube and either a pack of agar ordered from a chemist, or a standard 1 pint or 500 milliliter pack of gelatin, or even a standard 1 pint pack of dessert mix if it contains a gelling agent like agar, carrageenan, or guar gum. Dissolve the stock cube in 15 grams or a half ounce of agar, or all the gelatin or dessert mix into half pint or 250 milliliters of boiling water. Simmer for 30 minutes. Vegetable stock cube. Always be very careful with boiling water. Gelatin or agar. Sterilize the dishes and lids by boiling them quickly in water. Leave them in the water to cool. Standard size petri dish. Pour a shallow layer of jelly into each dish, put the lids on and leave the jelly to set. Then store the dishes upside down until you need them. This stops water caused by condensation falling on the jelly. Fingertip test. Press a clean, washed fingertip onto the surface of the jelly in a dish. Put the lid on the dish and leave it for a few days in a warm place. After this time, you should see pale, whitish, or creamy blobs, bacteria colonies, on the jelly in the outline of your fingertip. To look at these, you can either remove the lid and put the dish on the microscope stage, or cut out a sliver of jelly with bacteria on and place it on a slide. Before you do anything, though, and as soon as you remove the lid, you must carefully dribble some disinfectant over the bacteria and then leave this for a few minutes. When viewing, try both top and bottom lighting. Top lighting will probably be best. Press finger down on jelly. Outline a fingertip after a few days. Bacteria grow where your finger left food for them. Take care with knives and scalpels. Slice of jelly placed on slide. You should see one or more colonies. They may be different shapes.